I'm really pleased to have the opportunity to speak to you today and tell you a little bit more detail about the program that you've heard about from Stan and from Sarah today called Silence ALS, and tell you what we are learning from this program and what we hope to learn in the months and years to come. So in 2024, ALS, amyotrophic lateral sclerosis, is still a fatal neurodegenerative disease, and it's a disease that primarily affects motor neurons in the brain and the spinal cord. But ALS is, of course, not only a motor neuron disease. It has a number of non-motor issues. A large percentage of our patients have some degree of frontotemporal dysfunction, and ALS is linked in many ways, clinically, pathologically, genetically, to a form of dementia called frontotemporal dementia, or FTD. In the United States, the prevalence of ALS is about 35 to 40,000. 40,000 people suffering from ALS at any one time. So why is it that we're talking about ALS at a conference focused on the nano-rare? And the answer really comes from an understanding of the genetic architecture of ALS. 90% of our patients, 85–90% of our patients, have no family history, no underlying genetic mutation, but in about 15% of cases, there is a known disease-causing mutation in one of a large number and growing number of ALS genes. And the answer to the question of why this is a nano-rare comes in a study of the genetic complexity of this disease. So over the past 30 years, genetic researchers, including Brian Traynor, who's here in the audience today, have identified a large number, dozens of genes in which which mutations cause ALS. This study began in 1993 with the discovery of SOD1, and it continues today. This is a graphic showing the time course of gene discovery, and the size of each of these circles represents the relative proportion of these gene mutations in familial disease. And you can see the largest of these, C9orf72, represents about 40–45% of cases in European populations of familial ALS. SOD1 represents about 10–15% of familial cases. But for most of these ALS genes, they represent a small fraction of the total, 1%, 2% of all ALS cases. But 1% of 40,000 is still hundreds of ALS cases, and that puts us beyond the category of the nano-rare by Stan's definition. So then how is it that these cases of ALS are nano-rare? Well, the answer there comes from an understanding that in each one of these genes there are multiple individual mutations that have been associated with ALS. This is SOD1, in which over 150 different mutations have been linked to ALS, and these are mutations scattered across the gene and the protein structure from N-terminus to C-terminus. In the case of FUS and TAR-DBP, two genes we'll talk about today, there are 50 and 40, respectively, mutations that have been clearly linked, pathogenically linked to ALS. And each one of these specific mutations represents a nano-rare form of ALS that is amenable, in many cases, to ASO intervention. And for this reason, ALS fits very well into the n-Lorem platform. And understanding this, and as we watch the technology develop and the resources emerge in n-Lorem, we very soon after the beginnings of n-Lorem established with the foundation an initiative we call Silence ALS, initiative to discover and develop individualized investigational ASO therapeutics for ALS patients with these ultra-rare, nano-rare pathogenic mutations. This, as you've heard, is a partnership between n-Lorem and the Eleanor Gehrig ALS Center at Columbia that I direct. And the idea really was to create an infrastructure that builds on n-Lorem's nonprofit precision medicine platform and combines that with our disease-specific expertise in ALS and leverages our clinical and research resources, not only at Columbia, but throughout the ALS community to treat this underserved population of ALS patients with these very rare genetic forms of the disease. Our goal is to treat patients as close to phenotypic or symptomatic onset as is possible with an optimized ASO. And our ultimate goal, I have to admit, is to treat patients before disease onset with some of the same technology. And our hope, as I've said in some of the previous sessions, is really to create knowledge and experience and efficiencies that will inform our understanding and approach to all forms of ALS and to the related disorder, frontotemporal dementia. So the work of n-Lorem really fits well into our own workflow at Columbia and in many labs studying ALS around the world. This is a very patient-centric research focus. It begins with the patients whom we study. We study their genomics, their genetics, their multi-omics. We gain insights from those studies, and we use those to build animal and cellular models of the disease. And we study those models to identify disease mechanisms and pathways and identify targets that we can then go after with therapeutics of all kinds, including ASOs. And because of the genetic complexity, as I've told you, of ALS, it really, I think, lends itself well to an ASO approach. So my first experience with ASO technologies began through our study of a form of ALS associated with mutations in a gene called FUS, fused in sarcoma. This is a particularly horrible form of the disease. These mutations cause early onset, pediatric and juvenile, in many cases, onset disease that is very rapidly progressive. And the first case I saw of this, I like often to use this as an example of the aggressive nature of this form of ALS. This is Eitan Jacoby, an 11-year-old boy whom lived a stone's throw from our medical center in northern New Jersey. And he came home one summer with some mild weakness that progressed very rapidly. He was found to have a de novo spontaneous mutation in the FUS gene, a so-called P525L mutation. It was known to be particularly virulent. And he progressed very rapidly over a period of 9 months and died just after his 12th birthday, the youngest case of ALS, I think, reported at the time. So this motivated me in my lab to begin to study FUS ALS, to build models to understand mechanisms of the disease. And what emerged from a lot of years of work really was this idea that FUS caused ALS through a toxic gain-of-function mechanism that was dose-dependent, and that suggested a therapeutic strategy by which lowering FUS levels with an ASO might actually have a clinical benefit. There was no n-Lorem at the time. N-Lorem was a twinkle in Stan Crook's eye, I think, at this moment. But we approached Ionis, of which Stan was CEO at the time, and Bob Brown and I called Frank Bennett, and they very generously shared with us ASOs from their preclinical FUS program that ultimately, long story, led to the development of an ASO called ION363. This was a gapmer ASO that was non-allele specific, meaning it didn't differentiate between the wild type and mutated forms of the disease, of the gene. And we did this as an N-of-1 approach to help a young woman, J.C. Hermstad, whom Stan mentioned earlier. This was a young woman, 24 years old at the time, whose identical twin sister, Alex, had died of P525 ALS about 8 years earlier. And Alex was symptomatic at the time that I met her, and this is before n-Lorem, before the FDA guidances that you heard about. We approached the FDA about treating JC with this N of 1 ASO, and though it took some time, we got an IND for this and began to treat her. I believe this was in mid-2019. She responded in many ways to this. There was a dramatic decline in both serum and CSF levels of neurofilament light, which is a marker of a neuronal injury, we believe. And she did show some clinical stabilization, albeit at a low level with a FRS score of about 17, which is still pretty low. But despite the seeming benefits of this, JC died of her disease, complications of disease in the early part of the COVID epidemic. We learned a lot from JC. We learned a lot from the post-mortem studies that she made possible through the donation of brain and spinal cord. And this made it possible for us to continue through a series of individual patients, single patient INDs, to treat now upwards of 20 patients through our expanded access program, including a young woman that Stan mentioned earlier. This is Anna, who was also a P525L carrier, onset at age 15. And we began treating Anna relatively early in her course, but in the first 10 months of her treatment, she continued to decline steadily. But then, remarkably, at about 9 to 10 months after initiation of treatment, she really made a remarkable functional recovery, and this was associated with a dramatic, more than 80% decrement in her levels of neurofilament light. And this and data from other patients provided the safety and efficacy data that enabled the fusion study sponsored by Ionis, which is now ongoing, 3 years, I believe, and counting. This is a phase 3 international registration study that was made possible, I think, by the investigator-initiated ASO study. So first patients were relatively lucky here. Lucky that their numbers were at the borderline, some might say below the borderline for commercial viability, but for many of these ultra-rare ALS patients, there is no commercial option. There's no Ionis or other company that's going to step in and sponsor an international study. So the only option really is, as we've heard repeatedly this last day, Silence ALS and n-Lorem. So this is the workflow of Silence ALS. This is very much an n-Lorem program like others. I won't go through this. You've heard this morning in detail about how this works. This, I guess, is a little bit different in that we, as a partner of n-Lorem's in Silence ALS, work to try to curate some of these cases and to manage families and some of the stakeholders in this. We find these families, we characterize them, we perform the long-read gene sequencing that's required for ASO discovery. We obtain the consents and the samples that n-Lorem needs to initiate the ASO design and discovery process, and we work very closely with n-Lorem to initiate that as quickly as possible. You've heard in detail this morning about the work that they do and do so well to develop an optimally tolerated lead ASO. In the process, we work with them to develop a treatment protocol, and we're trying very hard to use the same treatment protocol with the same endpoints and measures across the Silence ALS platform so that we can compare patients with different ASOs and different mutations and different genes. So we can learn, as I've been saying, across this platform. n-Lorem files the IND, and then we work with Lori and her team to initiate treatment. Most of these cases have so far have been at Columbia, but we are building a network of centers throughout the United States, working with partners throughout the ALS community to deliver these ASOs to our patients as close to home and as efficiently as possible. So our focus largely has been on symptomatic individuals, and we really focus, as Joe and others have said, on gene mutations that cause ALS through an ASO amenable mechanism that is usually a dominant mechanism associated with a toxic gain of function, dominant negative effect, and not in our case yet, but possibly aberrant splicing mechanism. And we have multiple programs. I think I heard from Sarah last session that there are 30-odd ALS patients, ALS programs now active in and through n-Lorem. These are focused on multiple genes: TAR-DBP, CHEHD10, profilin-1, and Nexin A11, and we have other rare, ultra-rare gene variants that are in our sights, and we're preparing for nomination to n-Lorem. This effort, as you heard from Stan earlier, has been helped largely. The funding, has been provided in the last year by the Urgent Network of the NINDS. This is a program that is funding 3 ASOs a year for the next 3 years. It is a great beginning. We hope for more. And it is I think, one of NIH's first investments in the n-Lorem program, and I hope it's the first of many. So just quickly, I have a few minutes left. We've focused on a couple of genes in the first efforts of Silence ALS. Largely, we focused on a gene called TAR-DBP that encodes a protein called TDP-43. TDP-43 is a major player in the pathogenesis of ALS. It was first discovered in 2006 as a major component of these neuronal cytoplasmic inclusions that are a pathological hallmark of ALS, present in 97% of cases, including C9orf72 carriers. It's also found in nearly half of FTD cases. And the significance of this gene, this protein in ALS, was confirmed 2 years after the first descriptions of TDP-43 pathology in ALS by the discovery of a large number of pathogenic mutations in the TAR-DBP gene by Chris Shaw's group and Guy Rouleau's group at MNI. And this really, confirmed the central role of TDP-43 in the pathogenesis of ALS. So this TAR-DBP was one of the first targets of Silence in 2020. I think it was maybe the first, if not second, IND that n-Lorem was able to achieve. This was a 40-year-old woman with a rare de novo mutation in this gene, and quickly TAR-D001 was an allele-specific ASO with 55x selectivity for the mutated allele, and we began treating our patient with this about a year after the beginning of ASO design, which was a remarkable timing. But unfortunately, she had progressed very rapidly in that 1 year, which is a lesson that we're learning, sadly, that the disease doesn't wait for the ASO. We only got a few doses into her, and she was no longer able to participate. But very fortunately, we have been able to reuse this ASO. We now have 17 or 18 TAR-DBP cases that have been nominated and accepted by the ATTC, of which 13 or 14 are still active. Some of these patients have sadly died in the course of the program. But we recently got a second IND, in case number 16 here, to reuse the same ASO that we used 3 years ago. And dosing for that patient is scheduled to begin next week. And we have 2 ASOs in late stage of development, near ready for lead ASO selection and GLP tox. So in the coming year, we hope to expand our TAR-DBP case into the clinic much further. Important point, in the course of this, what we've learned is getting in to our patients with an ASO as soon as possible is really critical. And one way to do this is to begin the ASO design and discovery process before symptom onset. And as part of the Silence ALS initiative, we are characterizing, sequencing, obtaining samples from asymptomatic individuals, 21 listed here. I think we have twice as many now who performed long-read sequencing for haplotype analysis and phasing to begin the design process and allow us to design ASOs that would help as many people who could potentially convert with ALS in the months and years to come. And this is something we're doing across these ALS genes. Another program that's just getting off the ground is focused on a gene called CHCHD10. This is a gene in which mutations called multisystem degenerative diseases, various mutations associated with different phenotypes, but two mutations, R15L and S59L, associated with ALS and frontotemporal dementia. And in this case, we've pursued a non-allele-specific approach based on the biology of this disease, and n-Lorem developed NL-CHCHD001. This is, again, a non-allele-specific ASO with very similar chemistries, and the first IND for this program, I think we received in April of this year, and the patient who's being treated at Mayo in Jacksonville by Dr. Oscarson has received 4 doses. We've treated a second and third patient at Columbia with 3 and 1 dose respectively, and another patient scheduled to begin dosing later this month. Exciting program, initial data coming out already, and it'll be, I think, exciting to see this. This is one of the programs that has an IND for N of few. After 2 single patient INDs, n-Lorem filed a IND to treat 7 additional patients, and that was granted by the FDA. And we hope as more CHD patients appear, we can amend that IND to include larger numbers going forward. So I'll end and just say that there are many goals. We've talked about them a little bit in the last session. What do we hope to do in the years ahead? Really, I'm excited about, as I said, scaling the initial efforts of Silence ALS to involve more patients, different genes, more mutations in these genes, and then also looking to grow the Silence ALS Research Consortium both nationally and internationally as resources and regulatory pathways appear. I think, as I said, that we have an opportunity to gain efficiencies as we scale that will lead to a reduction in time and costs and allow us to treat more patients. As we build, we will grow a library of these Silence ALS ASOs, and we'll have a greater chance of reusing these for patients with shared haplotypes and targets of these ASOs, and that will also create a lot of efficiencies in time. We are going to expand our focus on these pre-symptomatic carriers. I think doing a lot of the work shy of GLP tox and GMP manufacturing is a way to get into these patients much more quickly, and we have an opportunity to do that through the larger ALS research community. I think, again, because a lot of these gene mutations can cause ALS or FTD, we have an opportunity to extend Silence ALS into Silence FTD, and we're beginning to do that through our collaborators in the FTD community, and there's great efficiencies, I think, possible there too. And finally, again, I hope that we can apply the knowledge we gain through the treatment of these ultra-rare cases to the non-rare, the sporadic, garden variety forms of ALS, and I think this is because of the shared biology of the disease, and as we begin to understand more about these very rare forms of the disease and how these ALS genes relate to one another and to this shared biology, I think will have a lot to contribute to the larger ALS community. So thank you.